Hi everyone, welcome to my channel Pure Biology, and today I come with another video, and this is the topic. The first part I already explained about the translation, and this is the second part, which factors are involved in the translation process. The first, we have to understand what is the three processes that are involved in the translation process, that is the initiation, elongation and termination. Okay, and I will elaborately explain about this, but in the next video. So here just I will try to explain briefly that what is initiation. And here in the initiation, it involves a binding of the small ribosomal subunit to small ribosomal subunit to mRNA, and then is joined by the 50S subunit, and it involves the reactions that precedes the formation of the peptide bond between the first two amino acids of the protein. Okay, if any sounds you can hear, so please you just ignore, because this place is just side of the road. Okay, because my place is just a side of the road. Okay, so please you just ignore the sounds. So what is called the initiation? Initiation means the binding of the small ribosomal subunits. Sorry. Okay, so what is that initiation? It involves the binding of the small ribosomal subunit. You all know that ribosomes has the two subunits. One is the large subunit, another is the small subunit. And the small subunits that bind to the mRNA and then is joined by the 50S subunit, and it involves the reaction so where the formation of peptide bond is occurred just between the two amino acids of the protein. And the second part, that is the elongation, and it includes the all reactions from the synthesis of first peptide bond and the addition of the last amino acids. Okay, here the elongation process explains the reactions or the steps where how the first peptide bond and the last amino acids, all the synthesis occur. That means synthesis of peptide bond and the addition of the last amino acid. This is actually explained in the elongation process. And the last one, this is the termination process, where you can see that the steps where the complete peptide chain that is released or the dissociation of the ribosome from mRNA. That means to stop the translation process. Okay, so here some initiation factors, some elongation factors, and some releasing factor. These are also important for the continuation of the translation process. So initiation factors means IF1, IF2, IF3. These three are very important. And the IF1 that binds over the A site of the small ribosome unit and subunit, and to prevent the initiator tRNA from binding site to the A site. What is saying that? That means it binds over the A site. That means it binds where? On the A site. Okay, on the small ribosomal subunits there are three sites, A, P and E site. So IF1 that binds over the A site, and in that way it prevent the initiator tRNA to bind with the A site. Okay, and second is the IF2. It directs the initiator tRNA to its correct position in the initiation complex to continue the initiation process. And then, and obviously here the GTPase activity is very important, because without GTPase activity IF2 cannot activate, or you can say its function is not occurring. And last one, this IF3, it binds to 30S subunit and prevents the premature reassociation of the large and the small subunits of the ribosome. That means there are the two subunits, large and the small. So here what is the function of this IF3? It prevents the premature, not mature, premature reassociation of the two subunits. Okay, and it also controls the ability of the 30 subunits to bind to the mRNA. That means it helps to bind with the mRNA, which it helps the 30th subunit to bind with the mRNA. Okay, and obviously it ensures the initiation site selection. So these are the three functions, and in the next video we will learn how this IF1, IF2 and IF3 they function in the translation process. Okay, here just their functions briefly we understand. Okay, elongation factors EFTU, EFTS and EFG. So EFTU, obviously you can say this is EF1A. So EFTU that directs the next tRNA to its correct position in ribosome. Okay, it directs the, okay, sorry. So EFTU that directs the next tRNA to its correct position in ribosome. Okay, where the correct position is very important, where that directs the tRNA, next tRNA, that means first tRNA and the next tRNA. So it directs the next tRNA to its correct position in the ribosome. And EFTS that regenerates the EFTU, okay, after hydrolysis of attached GTP molecule. That means GTP molecule when hydrolyzed, then it regenerates again EFTU. And the EFG, what is the function of the EFG? That mediates the translocation. Okay, so these are the functions of the elongation factors. And the release factors, there are also RF1 and RF2, and RF1 that recognizes the termination codon, that is UAA or UAG. So it recognizes the two termination codons. One is the 5' UAA 3' and the 5' UAG 3'. RF2 that also recognizes the two another termination codon, that is UAA also and UGA. Okay, 5' UGA 3' and 5' UAA 3'. And RF3 that stimulates the dissociation of RF1 and RF2 from the ribosome after termination. So there are the three factors. One and 2 both try to recognize the termination factor, but 3 that stimulates to dissociate the RF1 and RF2 from the ribosome just after terminations. So these are the actual basic explanation or brief explanation about the translation factors or the translation process. So I will try to explain how these factors and how these factors involved in this translation process, and obviously about the initiation, elongation and termination process we will learn. Okay, so please continue with my channel, and in the next video I will try, and so thank you. Okay.